Thank you everyone for coming here for the MCL workshop. Um, I'm inviting Dr. Andrew Stevens, to be the speaker for this event. He has had 10 years of experience in the development of smart medical devices for optimizing patient outcomes and streamlining medical care. And he is currently senior lecturer at Queensland University of Technology in Brisbane, Australia, and where the MCO work is conducted. Um, yeah, Andrew, I'm going to pass it over to you. Thank you for agreeing to do these slides.
Thanks, Josh. All right. Hi, everyone. Today we're talking about mock loops for cardiovascular device testing. Why should you listen to me? My background, I have an undergraduate in electronic engineering with no prior biomedical experience. How did I end up in this field? Mostly by accident. The reason I'm telling you this is because if you're an electrical engineer or a mechanical engineer and you really like this kind of stuff, you too can build a career in the wonderful world of biomedical engineering without having that specific degree. I get this kind of question a lot from my undergraduate students. So my PhD was in sensors and controls of ventricular assist devices. So during my PhD, I developed a fiber optic sensor and control system. That's a little picture of the fiber optic sensor that I had. My current research is in AI for MCS and other cardiac devices, clinical training tools. You can see our little dummy down here with some 3D printed flexible lungs, smart cardiac devices, and MCS delivery. So we're talking about things like um, surgical implantation, how the patient is managed during MCS. This is the kind of research I'm doing at the moment. So my team is the ACE Lab team. We have a very simple, basic motto. We solve doctors' problems using engineering. I suppose officially you'd call us a biomedical engineering lab, but we have lots of people from lots of different disciplines working in our lab, mechanical engineers, electrical engineers, mechatronic materials, and even occasionally we have clinicians come and do their research in our lab in amongst the engineers. As I said, we work on a range of things: artificial intelligence, CFD, emergency medicine, smart cardiac devices, valvular heart diseases, and also clinical training. All right, medical devices. Why do we need a mock loop? We need to evaluate our medical devices before putting them in humans. So, medical device regulation began in 1938 in America. The FDA is the most mature of the regulatory bodies at the moment. Since then, we've had many other regulatory bodies um, sort of pop up over the years. So currently we have the EMA in the EU, we've got the TGA in Australia, NPMA in China, PDMA in Japan. No matter what country you're living in, it's likely that the use of medical devices and drugs in people in your country is regulated by a regulatory body. That means it can't be sold, it can't be used without their approval. Why do we get this approval? It's for safety. So it's for safety of our patients. Obviously, we want to make sure that the risks of the device outweigh are less than the benefits. It's also for safety of business. So if your device actually kills somebody, there needs to be certain checks in place that says, well, we went through all this rigorous process to be able to get to a point where we say that we can safely put it in people so that when they sue you, you can point to that entire body of evidence and say, look, it was regulated at past regulation. That doesn't necessarily mean you're not at fault. It's just evidence to show that you did your due diligence. The most important thing, though, is it's used for transparency and patient confidence. So our whole medical construct is built on the fact that you can go to your doctor and you can trust what they prescribe to you. You can trust that they know what they're doing and that any device or any drug that they tell you to take is safe. We've seen in recent years people not trusting their doctors, not trusting the science, and we see how that can affect healthcare as a whole wider industry. And it has significant effects within the wider community. That's why, I really think that's the main reason why medical device regulation is required. It's for trust for the average person. So the typical purview of a regulatory body is looking at efficacy, so ensuring the drug or device works and that its benefits outweigh its risks. That it's safe. Not only does it benefit the patient, but it's actually safe to be used. Quality control, so that your device is consistently manufactured in a systemic and high quality way. You can't just give the FDA one device to look at and say, this is the standard for all devices. You need to have systems in place to ensure that everything is consistent throughout your whole manufacturing process. And also long-term monitoring. So even once you do get market approval from your regulatory body, they will require you to regularly submit reports about the performance of your device, any edge cases, and any unexpected adverse events that have happened throughout your device's time on the market. So it's not just a one timestamp, okay, it's a continuous process. Has regulation gone too far? Undoubtedly, the implementation of regulation has slowed down innovation particularly in our space. So to commercialize an artificial heart, you're looking at potentially hundreds of millions of dollars in clinical testing to be able to generate the regulatory evidence required to get a market approval for these devices. So I think there's a recognition in these regulatory bodies at the moment that it's too difficult. That kind of money can only be raised really by, large global multinational corporations, and that really stifles the market's innovation, but it also creates monopolies which make healthcare access difficult. The regulators are trying to find ways to streamline and reduce some of the red tape, but I think there's still some balance to be had between safety and innovation and moving the field forward. What kind of medical device development tools can we use? So it varies, but it can include in silico, which is computer-based models. Here we can do many simulations to try and optimize towards an operating point. We can use in vitro, things like benchtop testing, where we can take lots of different iterations and test them in a physical system. Benchtop testing is not only mock loops, we'll talk about some other things in a minute. In vivo, so we can do animal trials, including acute animal trials where you just put the thing in and see that it works, and chronic animal trials to see how it interacts with the biology of the body over a longer period of time. And of course, clinical trials in people eventually. And all of these types of evidence can be used in regulatory approval. So they all have their place in the development life cycle. Some in vitro models. Here we've got PIV, particle imaging velocimetry. That's a method of particle tracking in fluid motion that allows us to visualize where flow occurs. Here we've got a set of bellows that we can use for artificial heart valve testing. So these bellows generate thousands of beats per minute of the heart valve in order to try and simulate the whole 20-year lifetime of the valve in a very short amount of time, several weeks. So that would be accelerated mechanical testing. Here's a silicon ventricle that we can use for creating mock loops where we're interested in how the device fits in with the anatomy of the heart. Here's some tissue culture, cell cultures, which is definitely part of in vitro testing. We can take animal organs out of the animal hearts out of the animal, put them on a ex vivo rig, and implant our device directly into that animal's heart and see how that performs hemodynamically. We can also use pulse duplicators, which are essentially very basic mock loops that allow you to test your device under pulsatile conditions. But we're really here today to talk about mock circulatory loops. So a mock loop is a mechanical representation of the cardiovascular system. Essentially, it's a bunch of pipes and tubes, and each pipe and tube represents a part of the cardiovascular system, modeled by something we call the Wynn-Kessel model. So each of these chambers here is a compliance We've got left ventricle, left atrium, right ventricle, right atrium. We've got some little valves in here. We'll go through all of these components in a minute. A mock loop is what we call a lumped parameter model of the circulation. And what that essentially means is rather than modeling the flow path for every individual capillary, branch artery, branching vein, we take all of the capillary bed and all of the arterial bed, lump it together and pretend like it's a single sort of black box. And then we reproduce some of the physiology of those sort of lumped parameters. So the types of things we reproduce are the compliance, resistance. We'll talk about them together. But essentially what this does is allow us to group a bunch of things together to be able to simulate them physically. Advantages of a mock loop is it's cheap compared to animals. A mock loop can cost you anywhere between a couple of $100 to $150,000. That's still a lot cheaper than doing a lot of work in animals. Mock loops are highly repeatable, highly controllable, and easy to instrument. So when you're developing your device and you want to be able to see how it works under different conditions, it's very useful to be able to control a lot of different parameters, which is something that you can't do in the animal. You can't necessarily control for any confounders that you're seeing as you're testing your device. You can also get a lot more sensor information out of a mock loop because you can put more sensors in it. It can be very difficult to put more sensors into a person or into an animal. So it really is an essential tool for device development. Disadvantages is that it can't model all of the biological processes yet, although there's a lot of good research going on in that space, and that you've got to really build your mock loop for your specific research question. So what's the Windkessel model? The Windkessel model is essentially a mathematical representation of blood pressure and flow through vasculature. So through the arterial system and then returning back through the venous system. If we look at the elements 1 and 3, we have arterial resistance and arterial compliance. So that is the resistance and compliance of all of the aorta, all of the capillary beds, and all of the sort of branching arteries that come off the aorta. And we'll talk about resistance and compliance in a minute. And we also have the venous resistance and compliance. So that's all of the venous system that goes from the capillary beds all the way back to the vena cava and then returning into the right atrium. The fifth element is inertance, which talks about the acceleration and deceleration of blood flow, and we'll go through these as well. The anatomy of a mock loop, here we have each of the major arterial and venous beds represented by these large chambers. So this side of the system represents the body and organs. So you can see blood coming out of the ventricle into the aortic compliance chamber through the systemic resistance, into the systemic veins. So it goes through the capillaries, back through the veins, and then it gets returned to the lungs, where it will go through the right side of the heart. This side of the mock loop represents the pulmonary artery, so it gets pumped out of the right side of the heart into the lungs. The blood gets reoxygenated, returns to the pulmonary veins, and then returns back to the left atrium. We have the left and right side of the heart, including the left atrium, mitral valve, left valve, aortic valve, and the right side of the heart, right atrium, tricuspid valve, right ventricle, and pulmonary valve. So how do we actually mimic the pumping of the heart? Well, there are many ways that we can do that. One of the methods of actuation is pneumatic. So you can see the picture in the center top there. Our mock loop that chamber is filling up with fluid. And once it gets to a certain point, we shoot a jet of air into that chamber that causes the chamber to compress. And because we've got valves in the chamber, it causes the blood to flow in the forward direction. So you can see that's just kind of filling, ejecting, pushing down, filling. That's a relatively simple way to do it. You can use linear or hydraulic systems. So you can see on the right, or you can see on the bottom left, there's a silicon heart inside a clear box. And then there's a linear stage that's actually pushing and pulling fluid into that clear box, which is then contracting the ventricle, allowing it to fill and empty. Here's a rotary system on the top right. It's literally a motor connected to a camshaft style system where a piston just pumps in and out, and then a diaphragm transfers the pressure from the piston into the ventricle. And more recently, we've seen some physiological ways of making hearts beat. So this image in the center bottom is a little soft robotic sleeve, and when it can be wrapped around a real heart, or it can also be wrapped around a silicon heart. And every time it compresses, it kind of recreates that torsional motion that the heart actually does when it's beating. So there's lots of different opportunities there to implement your atrial or ventricular contraction. Another really important part of your mock loop is your valves. So there are a wide variety of valves out there. You can use a check valve like this, where this lid opens up when there's enough pressure to push forward. You can use balloon valves, duckbill valves, ball check valves. The valves that we use in our mock loop are actually polyurethane heart valves that we've designed ourselves that have the correct anatomy and correct opening and closing of the aortic valve, a tricuspid valve. The most important thing to consider when you're choosing your valves for your mock loop is cracking pressure and the healthy pressure drop. So cracking pressure is how much pressure does it actually take to build up in this chamber. Imagine this pressure zero to overcome the resistance of the flap, whatever your valve's made of, and open it up. And you ideally want that cracking pressure to be less than 1 millimeter of mercury, which is what it is in the native valve. So the native valve itself, it offers very little resistance to opening and closing. The other thing you've got to consider is the pressure drop. So how much loss of pressure do you get as the flow travels across the valve? So in a heavy valve like this, you might get 10, 15 millimeters of mercury drop. In a valve like this, you might get 5 to 10. In a big ball valve like this, you might have a very high cracking pressure and a very high pressure drop because it's actually got to travel around the ball through these small channels. You ideally want your pressure drop to be less than 5 millimeters of mercury because this is within healthy ranges. If your pressure drop is too high, rather than simulating a healthy valve, you're actually simulating aortic valve disease or any kind of stenotic valve disease. This means that on top of your diseased patient state that you might be simulating, you'll also have a second disease state, which you might not realize you have. The other thing that we talk about in terms of mock loop properties is compliance. So compliance is really the stretchiness of the vessels. When your heart ejects the fluid, the blood flow travels as a pulse wave down your vessels, out of the aorta and down all the way to the capillary beds. So the blood comes out, the vessel actually stretches due to the change in pressure, and then it sort of springs back, and that helps to propel that pulse down the arm. Mathematically, we think of compliance in terms of change of volume over change in pressure. So how much pressure does it take to push this amount of volume into the chamber? And this is really related to the elasticity and the springiness of the vessels. Another way to think about it is if we have a column of water with some trapped air in it, and we force volume into this column, that water column is going to move up and compress the air. The compressed air then acts like a spring, giving resistance to pushing more water in. And then when that spring releases, it forces that water back out. So there are several ways to implement compliance into your system. You could build, for instance, tubes that have the same compliance as the vessels that you're trying to replicate. We personally use passive compliance chambers. So a passive compliance chamber is either a closed to air chamber with trapped air inside or an open to air chamber with a specific diameter. It's worth noting that the difference in arterial and venous compliance is quite high. So the venous compliance is about 20 to 30 times higher than the arterial compliance. And this means that the veins are 20 to 30 times more stretchy. So high compliance means stretchy, low compliance means stiff. You'll notice in this one here that the compliance is a curve. And that makes sense, right? Because when you're stretching out these vessels, they sort of get to a certain point where they can't stretch anymore. And then as you try and increase and force volume into those vessels, the pressure goes up rapidly. So that's why you can see it kind of goes up quickly and then plateaus off. For aortic compliance, you're looking at a compliance between 1.1 and 1.5 mils per millimeter of mercury. Pulmonary artery compliance, and that's this chamber here, by the way. Pulmonary artery compliance is the chamber behind it. That's 3.5 to 12 mils per millimeter of mercury. Systemic venous compliance is about 100 to 200 millimeters of mercury, so it's huge. That's represented by an open to air chamber where all your compliance in an open to air chamber is just a single value. Because what that means is the only resistance you have to volume, to pushing volume in the chamber, is the change in hydrostatic pressure as that chamber gets fuller and fuller and more pressure pushes down, opposing volume coming in. Our pulmonary venous compliance, which is this little one back here, is about 7 to 12 millimeters of mercury. You can also implement what's called an active compliance chamber. So an active compliance chamber is varied by actively changing the pressure in the chamber. So this one here, air can be blown into the chamber either to a fixed level or it can be changed during the heartbeat to do waveform shaping of the aortic waveform. And air can be released through this port here to reduce the volume of trapped air in that chamber. Similarly, this one's a pusher plate with a diaphragm where they're just physically changing the amount of trapped air and the pressure that the trapped air is pressurized under just by moving the diaphragm in and out. So depending on what you want to look at, active compliance chamber might be really important if you're looking at aortic pressure waveform shaping, for instance. So resistance, our vessels can expand and contract based on the needs of the body, based on the blood flow requirements, and based on the amount of oxygenation that the end organs need. So as you probably know, when the vessel is dilated, blood flows more easily, there's less resistance. And when the vessel's contracted, blood flows less easily and there's more resistance. Mathematically, we define resistance as the difference in pressure between 2 points, always between 2 points, over the flow. Resistance is measured in either Wood units or dynes per second. And for those of you who are paying attention, this is just Ohm's law. V equals P on Q. The simplest way to implement vascular resistance in your system is just a tap. You can see this is where our vascular resistance for our systemic side is. This is where our vascular resistance for our pulmonary side is. A more sophisticated way of doing it is using either a hydraulically, pneumatically, or electrically activated pinch valve, where you can just with a click of a button or entering a value, increase and decrease the resistance dynamically. And the benefit of this is that you can do it very repeatably. You don't have to get up from your computer and then go physically manually change something. And you can just use simple PI control to set a specific SVR. And then no matter what you change in terms of pump speed or ventricular contraction, the controller will maintain this specific SVR. So inertia is the other element we talk about. Inertia is about the acceleration of the fluid and how much force it takes to accelerate and then decelerate the fluid. Mathematically, we talk about it as the change in pressure over the change in flow with change in time. For a straight rigid tube, this can be simplified as rho, the density of the fluid, multiplied by the length of the tube over area. So the way that we simulate this in our mock loop, one of the key important elements is choosing a working solution that has a similar density and a similar viscosity to blood. The easiest solution that we use is water, a mixture of 60% water and 40% glycerol by weight, not by volume, which at 23 degrees Celsius, lab temperature, has a viscosity of 2.6 centipoise. This is the same as human blood at 37 degrees, which has a viscosity of 2.3 to 4.1 centipoise. The density of glycerol is like 1.006, and the density of blood is, the density of blood is 1.006. The density of water is about 1. When we mix this solution together, we get a very similar density. The other thing to consider when you're having inertia in your system is how far away the physical elements are from each other. So obviously the left atrium and left ventricle in your heart are extremely close together. If you've got your left atrium here and then you've got a big valve in here and you've got your left ventricle, then the time it takes for that fluid to move from one chamber to another will not be the same as in a heart. So you generally want to keep things as close together as you possibly can. You can also add some very cool advanced features to your mock loop. So you can have a Starling mechanism, which automatically adjusts heart output based on the requirements of the body. You can have a barrow reflex, which adjusts resistance and heart rate to maintain perfusion pressure. You can have coronary regulation, which maintains a constant blood flow to the heart, or cerebral regulation, which maintains a constant blood flow to the brain. All of these can be mathematically modeled for healthy and disease states. So you take data from your sensors and then you feed it into different actuators to change things like the compliance, the resistance, and the flow through each system. Lastly, I'll talk about hybrid mock loops. So a hybrid mock loop is a mix of physical components on the bench and mathematically simulated components in the computer. So you can see in this example here, there's an actual heart on the bench there. And then there are a couple of pressure reservoirs which are changing the pressure that go into and out of the heart. And then you can see there's a bunch of sensors that go into a numerical model, which are simulating what's happening in the lungs. And so based on what's happening in the lungs, based on the simulations and based on the sensor feedback into the model, this adjusts the pressure before and the pressure after the heart in those reservoirs. So the benefit of having a hybrid mock loop is that you can reduce hardware space and cost and accurately simulate individual compartments. So remember when we were talking about how our lumped aortic compliance is our aortic artery, as well as all of our capillaries, all of our sort of arterial branches that come off the aorta. If you wanted to, you could simulate all of the different compartments and how blood flows distributed through those different compartments in the computational side, and then feed that back into your mock loop so that you can get a more sophisticated approximation of the blood flow through the different aortic compartments. It really depends on what you're looking at. Another really popular thing is to use hybrid mock loops to simulate oxygen and gas exchange and simulate biological and autoregulatory processes. This, of course, comes with some limitations like requiring some pretty powerful compute power. When you build your mock loop, it's really important to validate it. So at a minimum, I would say that you need to validate your mock loop using waveforms, averages, and ranges based on the literature and based on the disease state that you're trying to replicate. So most of you will be trying to start with a heart failure condition and then put a pump in and see how that pump improves that heart failure condition. You can use prospectively collected data if you have the capability. If you're interested in geometry, you can use something like PIV to ensure that your flow dynamics are what you expected. And you can look at the repeatability and response of your mock loop. So you can look at the time response, overshoot settling time, and how repeatable your mock loop is from one experiment to the other. Mock loop should be highly repeatable between each experiment. You should always validate based on what you're specifically looking at and the design of your artificial heart pump. Data acquisition and control. So depending on complexity, you're almost certainly going to need some pressure sensors, flow sensors, volume sensors. So these are some clinical pressure sensors. You can use you can get pressure sensors anywhere from a couple of bucks to a couple of $100 to possibly even thousands of dollars. With your pressure sensors, make sure that they are compatible with liquids because some of them are only compatible with gases. Flow sensors, there are a range of flow sensors, including clamp-on, ultrasonic, paddle wheel, electromagnetic sensors. Volume sensors, there are ones that can use a float or lasers or ultrasound. Really depends on your mock loop design and your budget. For the brain will be taking in sensor readings. It'll be recording data from your mock loop, and it may be controlling actuators like your systemic vascular resistance. So, and also your heart's actuation time, its pumping time. So the brain can range start at a D-Space, which is about $28,000. It's a top of the line machine. National Instruments is about $6,000 to $12,000. LabJack's less than 1,000. And these ones here go from a couple of $100 down to $20, $30. Important things to consider when you're picking your data acquisition. Make sure you're comfortable programming it and make sure it's got enough resolution to read your sensors. All right, so TAR, do I need all of this if I'm developing a total artificial heart? For a pulsatile total artificial heart, yes, you'll need most of this stuff. You won't need ventricles and atria because obviously you're going to have to take the whole heart out to put the artificial heart in. You won't need valves in your mock loop, though you may need valves in your pump itself. For continuous flow TAH, you can get away with even less. So you probably won't need a sophisticated control system. You might need compliance. Keep in mind, compliance only affects the hemodynamics when there are positive and negative changes in over time. So for instance, if you speed up and slow down the pump, the compliance will affect what happens. If you're just letting the pump run at a constant speed, the compliance isn't going to make any difference. It's only for transient changes in blood flow. Resistance, you will absolutely need resistance in your mock loop, and you will absolutely need a pulmonary and systemic circulation, both of which probably you will want to be physical circuits. All right, summary and final thoughts. So mock loops are a mechanical representation of the heart and cardiovascular system. Build your mock loop based on your individual pump and your individual needs, and configure it in a way that will easily connect to your pump. Calibrate your mock loop, validate it, and recalibrate it often because it's prone to sensor drift. Pay particular attention in validation in the areas that you're measuring. Big tip is to buy extra of everything because it will break, crack, get damaged, or blow up. And you want to just have some spare parts on hand so that you can swap stuff out, especially when you're in crunch time trying to get data for your report. Consider modularity. So if you glue your whole mock loop together and you've got a big system like this, how are you going to transport it, break it apart, and put it in your suitcase? So consider how you can get it together and apart for travel, and be prepared for leaks because it will leak often. So with that, I'm going to say good luck in building your mock loop. If you want to learn more about mechanical circulatory support, this book here includes everything from heart failure, building and designing artificial hearts, testing artificial hearts, all the way to regulating artificial hearts and clinical commercialization of artificial hearts. I would encourage you to go out and purchase one, at least as a team. If we get enough sales, they'll let us write a third edition, which I would be really excited for. So that's it. Thanks from me. If you're interested, reach out to me. We do offer some internships, some research internships. Unfortunately, they're not paid, so hopefully you've got rich parents. This is our lab website. And yeah, thanks for showing up.
When making a pulsatile related total artificial heart, and does the mock circulation loop is it preferred to have a like a water with glycerine to have analog blood, or is it fine to use water to kind of simulate the um, in vitro
Yeah.
study?
Yeah. You need water and glycerol for both pulsatile and continuous flow for different reasons. The pulsatile VADs, the water, the solution will affect the inertia of your pump. So because the water glycerol solution is more viscous than water alone, it will actually require a lot more force to push your blood out of whatever sac collector thing that you have, right? So if you just test it in water and then you have to move to blood, you're going to have all these additional friction forces in your pump, means it's going to be actually working much harder than what you thought it was going to be working in. For continuous flow, the viscosity of the pump of the working solution hugely affects the pressure flow characteristics. So you really need to be using a water glycerol or some other blood analog as you're testing and designing, so that when you do get to blood, you're not like completely way off your operating point.
Nice, thank you. Also, would like to mention to everyone, you could also like ask some questions on the chat as well. So, yeah.
Yes, I would like to know how necessary it is to simulate a non-Newtonian interaction with the blood.
Now this is a great question. So for those of you who don't know, blood is a non-Newtonian fluid. And what non-Newtonian fluid means is its viscosity changes depending on what shear it's exposed to. So things like water, their viscosity don't change with shear. They change with temperature and they change with a few other things. Because blood is made up of plasma and also red blood cells, when you actually put those red blood cells through a pump, they shear and they become more elastic and they become more stretchy. Sometimes they even rupture, and that actually changes the viscosity of the overall blood content. So the question is, how important is it to test with non-Newtonian fluid when you're developing your device? The answer is somewhat. So you can get really a lot of the way through your testing using Newtonian fluids like water glycerol solutions. The really important thing that you can do is once you've got your design finalized, you will actually at some point need to test it in blood to look at hemolysis, right? And so as a part of those hemolysis tests, then you can also compare the performance of your pump as a function of, or as a secondary objective of doing those hemolysis tests. So in short, I wouldn't necessarily do it just for the sake of doing it, but it's something I would keep in mind when I was preparing my hemolysis tests. So the reason I say don't bother doing it is because it's not that easy to get non-Newtonian fluids that behave like blood that aren't blood. It's not that easy and it's not that cheap either.
Yeah, thank you.
I thought Jessica unmuted for a second. Are you going to be brave, Jessica?
Yes, I will be brave.
Please.
Um, our team has been transitioning to using more STM32s. Is that okay for us to use for the mock loop?
Which team is that?
Mending Broken Hearts.
Mending Broken Hearts. Okay. What the hell is STM32? That's your microcontroller.
Yeah.
Yeah, okay. So the main limitation you'll find of your STM32s is probably your analog to digital converters. So I think do you know what the resolution of those is? I think it's 10 bits.
Unfortunately, no. I'm a mech student, so
Yeah. So in more accessible terms, what you might find with those controllers, if you just try and read an analog input from them, is that your pressure and flow waveforms might look a bit like jagged because they have a very low resolution. The best way that you can get around that is to use like a little shield or something that has an analog to digital converter in it with a very high bit rate. So you want kind of like 16 bits so that when you're reading the voltage out of your pressure sensor, it converts that to a nice, smooth pressure waveform, which you can then read into your sensor. The other bottleneck you might have with your STM32 microcontroller is that they're not super powerful. So depending on how much data you want to record at a single time, you might find that you reach an upper limit of how many operations they can do. If you want something that's not too expensive, that is quite a step up, I would recommend the Arduino Due. They're about 100 Australian dollars, but they have quite a lot more power. The only difference between them and your other STM boards is that they're all 3.3 volt. So you'll have to be really careful not to put anything 5 volt on it or you'll blow it up.
Yep, that sounds good. What about in the case that we can spend a little bit more money? For example, like they suggested on the slides.
The next one up I would go is probably the lab jack. Like if you had like say $600-700, the lab jack. If you're kind of somewhere between $100 and $700, I haven't really operated in that area. So I couldn't tell you 100% how, like, what is good out there. Some people like Raspberry Pi. I think Raspberry Pi is better as a computer than as a data acquisition system, though.
Yeah. All right. That sounds good. Thank you.
My pleasure. I've got a question on chat. We're looking into using a PI controller. How do you recommend approaching this? And in your experience, how difficult is it to implement? Good question. Depends on how much control system theory you've done. If you're really interested in learning and you have access to MATLAB, there's quite a few good both MATLAB and Simulink tutorials on implementing baby's first PI controller. In terms of physically implementing it, it's actually super duper simple. If you're thinking about it in terms of C code, your proportional component would just be, where am I now? Where do I want to be? And what's the difference between that? And then add a proportional gain to that. And your integral component is, where am I now? Where do I want to be? And how different was that from the last step that we had? So definitely there's some really good MATLAB tutorials out there. And as with everything, if you just trying to get your head around the theory of it, there'll be a guy on YouTube, probably an Indian guy, who will explain it so well, like unbelievably well, that you just get it. Is there anyone here today who's not from Australia? Did anyone wake up for this? NZ, that's great. Is it recommended to design your own controller, or is it a bought motor controller sufficient? It's a good question. At the outset, I would say try and get as much stuff off the shelf as you can so that you don't have to reinvent the wheel. So like if you're using, say, a brushless DC motor as your foundational architecture, just get a brushless DC motor driver off the shelf so that you can then just say maybe put a voltage in to change the speed, right? Once you've got that thing working, then you can start to work on miniaturization and whatever feedback sensing you need in your system. So I would say start off the shelf. And then once you reach the limits of what that off the shelf thing can do in terms of being able to, in terms of what it can technologically do, in terms of how small you can make your package, that's when you can then start to work on building your own stuff. But yeah, just honestly, get your first prototype up and running with as much stuff off the shelf as you can. Oh, this is actually a really good one. So this is not my area of expertise. As I said, I'm an electrical engineer, so my mechanical engineering expertise is quite limited. The question is around designing a small scale portable model and key differences in terms of physiological parameters, accurate scaling, things like that. So, in terms of the electronics, there shouldn't be any major differences between a full scale and a small scale model. But in terms of the hydraulics, I think the differences will be huge. So, if you're say you've got a full size pump and then you reduce it by 50%, then your whole operating point will change. Your HQ curves will change. You would actually need to experimentally validate what your miniaturized pump's HQ curve looked like. And then you might actually need to change the physical properties of that pump to then go back to your full-sized operating point. So what I mean by that is, say you've got a rotor that has a certain blade, it has a certain blade gap, so many blades, so many angles, and that produces 100 millimeters of mercury pressure head at 5 liters a minute at 5,000 RPM. If you then shrink that thing down and you still want that 100 millimeters of mercury at 5 liters a minute, you will need to probably completely redesign the rotor and also change the speed that it's operating at. The smaller the pump, the faster the speed needs to be because it's just not pushing as much volume through the system. Exactly how it will change, I can't say because it's not my area of expertise, but it will definitely be different. Definitely. No, the pressure sensors. So the question is, when purchasing pressure sensors, do we consider the thicker viscosity? Not at all. The pressure sensors will ideally operate using some kind of membrane. And so as long as something's pressing against the membrane and the membrane's impervious to liquids passing through it and getting into the electronics, it should work whether you're measuring oil, petrol, water, glycerol, tomato sauce. It should just work. That being said, when you're ordering flow sensors, you absolutely have to consider the viscosity because the viscosity will give you, or the amount of flow and the type of flow that you get through your through your flow sensor will depend on the viscosity. For the passive compliance chambers, the caps will be, we just make our caps either we 3D print them or you can literally just buy a PVC cap that sits on top. How do you go about calculating the elasticity of it as well as the size of the chambers? It's all about the volume of trapped air. So, essentially, what you need to do is you need to understand that the change in pressure will be rho GH, right? So it'll be the density of the fluid versus the gravity versus the height of the column, as will the change in volume. So you then need to work backwards from the compliance value that you want. You'll know rho, you'll know G, you need to calculate H, and you need to calculate how that will change as the compliance chamber fills up. If you send me an email, I can send you a master's thesis which outlines exactly how we calculated our volumes of trapped air. And I can probably also even send you the volumes. So we use 4-inch inner diameter tubing for our aortic and pulmonary venous compliance chambers. And then all we need to do is adjust the height so that when we fill our mock loop up to our 0 level, we know exactly how much air is trapped in the chamber. So there is a formula for it. I can't remember, but if you email me, I can send it to you.
Can I ask another question?
You can ask as many questions as you want.
Um, well, we're planning eventually to connect it to our own clinician website. I'm not sure if this is your area of specialties, but
Hmm.
would there be any complications with doing that?
So you,
Like
um,
to display data through numbers or maybe graphs?
is the clinician website just a public website? It's not like hosted in like an EMR or anything like that?
I'm not too sure. That's also another sub team, but the purpose of it is purely just for like clinicians to only look at it. The patients won't have access to it.
Yeah. So if you, it depends on how you implement it. So if you want to implement it within the hospital system, yes, bloody hard, like really hard to get data into and out of hospitals. What I would suggest as a proof of concept is just do like a public website where you can stream data from your, say, microcontroller or your Raspberry Pi to that website live so that like people could just log in publicly and see it. And then once you've just got that sort of uplink between the two sites, then you can like add some user logins and things like that and restrict who can and can't see it based on what login. So I'd start super basic, just streaming it to like a not even like a live site, just like a local host so that it's like simulating the website on your computer and then build it into a public live site and then just keep adding features and features and features. So it's a really great initiative. And technically, I think it's going to be reasonably easy. It will definitely help if you have a software engineer involved who knows how to do like front end, back end stuff because they don't really teach that stuff in engineering, biomed, or electrical or mechanical, right?
Yeah. Okay, yeah. That sounds like what we have now. I didn't really know, so I just want to ask you how doable it was. But yeah.
Oh, it's super doable. Yeah, yeah,
Yeah,
it could be
okay, cool.
done. Yeah,
Okay, cool. Another one for
please.
the diameter and length of the pipes that are going to be connecting between the chambers or
Yep,
between our heart.
yep,
How important are they?
yep. Reasonably important. You want them to be small enough. You want them to be small enough that you can connect the two parts together. You want them to be big enough that they don't massively impede flow and create unnecessary resistance. So I think we use either 1 inch or 2 inch. And then we actually cut them down so that the physical chambers are as close as possible together, basically touching. And you just use like, you use the offcut, you put some glue around it, you stick one end in, you stick the other end in, and then you put it together. Or if you want to make it more modular, you can use something like a cam lock. Yeah, 1 to 2 inches should be just fine.
Perfect. That sounds good. Thank you.
Yes, I have another question.
Yes,
Uh,
where are you
do you,
from?
I'm from Thailand.
Perfect.
Yes. I
Please.
have another question. I would like to know how do you simulate like thrombosis in your system? Is it like
Oh,
necessary? And
that's
how like other consideration other than hemolysis,
yep. Yeah, yeah,
those kind of things.
that's a great question. The answer is it depends where you want to simulate the thrombosis. So we actually, um, if you want to simulate it in your pump, what you can do is use like particles. You can have particles of a certain size that you can suck into your pump and then see what it does to your pump. And of course, it's going to be a bit random. One of the things that we tried before was gelatin, actually. You can use gelatin, basically aeroplane jelly, to create blood clots, artificial blood clots, by dripping gelatin into water, I think it is. There's been some research done on that, and essentially that has the same density as a sort of newly formed blood clot before the blood clot becomes fibrous and horrible. So that's one way that you could do it for simulating it in the pump. If you're simulating it, say, in the outflow graft, it might actually just be easier to create a restriction to see how it affects the hemodynamics. Because obviously, the major problem with thrombosis, except for stroke, is that it will affect the performance of your pump. So if you want to simulate it in your mock loop, again, you can put some actual physical things in there to clot up your mock loop. Those are all options.
And I have another follow-up question.
Mhm.
I would like to know, do you have a physical way to calculate shear stress in the pump?
Calculate shear stress in the pump. The first thing that we usually do is computational fluid dynamics simulations. So you would basically recreate the geometry of your pump, recreate the boundary conditions that are talking about the flow through the pump and the spinning of the impeller, and you would do computational fluid dynamics to calculate or estimate the shear stress of that's being imparted from the pump onto the blood. You can measure shear stress of blood using an actual setup with blood. I'm not an expert on it, but the team at the Gold Coast is an expert on that. Mostly we just start with simulations, and then when we look at how to validate those simulations so we can be sure that our results are correct.
Thank you.
I've got another question. If your setup has large resistance, can this be compensated for? Generally speaking, you want the setup of your pipes and stuff to be not so restrictive, because you can always increase resistance by using a pinch valve. If your setup has too much resistance because you've used pipes and tubes that are too small, it is impossible to then reduce your resistance unless you add more channels or a bigger pipe. So as I said, we typically use between 1 and 2 inch pipe for all of our conduits. And then you can it's easier to increase resistance than decrease resistance, if you get what I'm saying. That's a cool idea. I think if you so the question is it possible to make an MCL battery powered? I think if you made a battery powered MCL, you get a lot of novelty points. I don't think you'd be able to run it off like a single 9-volt battery. But if you had, like, say, even a 12-volt motorbike battery, I reckon it would be actually very possible, especially because in a total artificial heart mock loop, you're mostly just controlling the resistance and reading stuff from sensors. So I reckon you get a lot of novelty points if you could pull that off.
Yeah, awesome. Thank you guys for attending. Yeah, it's been a good session. Thank you guys.
Thanks, guys. I hope I see you at the competition.